In this video, we will have a brief review of cell structure and function. The plasma membrane surrounds the cell and provides a semi-permeable structure. The plasma membrane will separate the intracellular environment from the extracellular environment. Purposes of the plasma membrane include transport of materials in and out of the cell. On the cellular membrane are receptors for signaling. On nerve and muscle cells, it's the cell membrane that propagates electrical current, and the cell membrane regulates growth and proliferation of the cell. The cell membrane is a lipid bilayer made up of phospholipids with a hydrophilic head and a hydrophobic tail. The plasma membrane is insoluble to water, but because of this hydrophobic or lipophilic tail, it is soluble to lipids. On the plasma membrane, we have a number of proteins that serve different functions for the cell. For example, you might have a channel protein that will selectively allow certain materials through the membrane. This might be something like a glucose channel. We also have receptors on the cell that bind to a ligand or some sort of substance to cause an effect in the cell. We have proteins that allow for recognition of the cell by our immune system. And so all these proteins on the plasma membrane carry out specific and important functions. Our cells need to communicate to function, and so a simple cell communication system consists of a receptor and a messenger or a ligand. The binding of chemical messengers on the cell surface or inside the cell is called signal transduction. What happens is our ligand or that messenger molecule will attach to a receptor protein, and we can control the cell response by upregulating or increasing receptors or downregulating or decreasing receptors. So our ligands, like a hormone, are what we call our first messenger. These can include things like steroids, neurotransmitters, growth factors, protein hormones, different molecules that are going to signal the cell to do something. This ligand, this first messenger, will bind to a receptor, which are specialized for our cell type. We have different types of cell surface receptors, such as a G protein linked receptor, an enzyme linked receptor, or we could have an ion channel linked receptor. And we can also have intracellular receptors. These are going to be our lipid soluble ligands, such as thyroid hormone, steroid hormone. And these intracellular receptors bind in the cytoplasm. And what happens there is it leads to gene expression. So it turns on transcription to create a new protein. Our cell surface receptors have a pre-made response, and so they're going to be a much faster signaling because they're not making a new protein. Now, our cell membrane will regulate the substances that are inside of the cell. And so to do this, we have to have some what we call selective permeability. And so this involves transport of substances across the membrane. We have different types of membrane transport. These include diffusion, active transport, and vesicular transport. So diffusion is a passive process. In diffusion, substances move from areas of high concentration to an area of lower concentration. We call diffusion passive because it's not active. It does not require an input of energy. So diffusion does not require ATP. In diffusion, how quickly things move, so that rate of movement is determined by the relative concentration of substances on either side. There are some specialized types of diffusion. So facilitated diffusion is used for substances that cannot cross the lipid bilayer because of some sort of characteristic, like their size, their charge. So charged particles can't go through there, or polarity. Because remember this lipid bilayer, the tails are nonpolar. Some examples of substances that require facilitated diffusion include things like ions, glucose, and amino acids. And again, facilitated diffusion does not require ATP. It's just a special tunnel, you can think of it, that allows certain particles to move from a higher concentration to a lower concentration. Another specialized type of diffusion is osmosis. Osmosis is the diffusion of water across a semi-permeable membrane. Water is going to move from a higher concentration of water to a lower concentration of water. But a might be perhaps easier way to think about it is that water is always going to move where it's more concentrated. So think that water always wants to be where the party at. It has fear of missing out and it wants to be where all the solutes are hanging out. So here in this picture, where you have one side of the container has a lower solute concentration as compared to the other, with a semi-permeable membrane on either side, the water is then going to passively move to where there are more solutes through the process of osmosis. Now we'll talk about active transport. So unlike diffusion, which is a passive process, active transport requires energy. It requires ATP. And it requires ATP because we're moving solutes uphill. We're moving solutes against the concentration gradient. So if you think of passive as something rolling downhill from high to low, active transport is going to go uphill from low to high. A common example of active transport is the sodium-potassium pump, or you might see it as the sodium-potassium ATPase pump. This pump transports sodium out of the cell, and it moves potassium into the cell. So remember that in the extracellular space, it has a higher concentration of sodium as compared to the inside of the cell, and so we're pushing that sodium back into that extracellular space. The most abundant cation inside the cell is potassium, and so we're putting potassium back into the cell up its concentration gradient. During this process, we move 3 sodium molecules out of the cell, and we pull 2 potassium molecules into the cell, and again, we're going against that concentration gradient. Vesicular transport is where there's a vesicle or some kind of little vehicle that moves substances. So we have 2 types. We have endocytosis. This is where we bring things into the cell. The cell will ingest material by enveloping it in a portion of its cell membrane, and then it pinches off that portion of the cell membrane. We also can have exocytosis. This is taking stuff out of the cell. And again, we will create a little secretory vesicle that then transport the substance up to the outer edge of the cell, where then that allows the membrane to open up and dump it into the extracellular space. When we look at overall cell structure, we have different components. Within the cytoplasm, we have membrane-enclosed organelles, a cytoplasmic matrix, inorganic ions, most predominantly potassium. We have organic molecules such as carbohydrates, proteins, lipids, RNA, and we have water. And then within the nucleus, we have genome and the enzymes required for replication and transcription. And so we'll look at different organelles within the body. And so I like to think of the cell as a little city. And a city's job is to support everything within that city. So there's a lot of work that happens. And so our cell is divided up into different departments. The first department is the department of protein production. People who work in this department are in charge of protein synthesis and packaging. This includes our ribosomes, which contains nucleoproteins such as RNA plus proteins. And these are important in protein synthesis. We have an endoplasmic reticulum. Rough endoplasmic reticulum makes proteins, and smooth endoplasmic reticulum makes our lipids and steroids. And then packaging of proteins occurs through the Golgi apparatus. We also have a department of sanitation, so we have to clean up the cell. We make a lot of waste, like any city. And so we have different subdepartments within the Department of Sanitation. We have the lysosomes. These contain enzymes that break down and remove foreign substances and worn out cell parts. Our peroxisomes, so these have oxidative reactions that involve hydrogen peroxide. And so they help with reactive oxygen species and things. And they also produce bile acids in our liver. And our proteasomes break down proteins that are deformed or deranged in some way that are identified for destruction. And that process is called ubiquitination, where you tag a protein that says this has to go. And we have our Department of Energy. This is completely run by the mitochondria, which is the powerhouse of the cell. And mitochondria also regulates apoptosis, which is that programmed cell death. And that wraps up our brief overview of the cell.